Hello student, today we are going to discuss the two plants of the Paleobotany, that is the fossil plant. The first plant is called Lyginopteris and second one is Enigmocarpon. So first we go with the Lyginopteris. So Lyginopteris is the fossil plant and which is discovered from England as well as from America. So this Lyginopteris is also the organogenera or the form genera plant. That means the root of the Lyginopteris, stem of the Lyginopteris, that means different parts of the Lyginopteris fossil plant, they have separate generic name. So this is the plant, and this plant we have studied. First one, that is the classification of the plant. So the Lyginopteris plant is classified as kingdom Plantae, subkingdom Spermatophyta, division Gymnosperm, because the seeds are naked, class Cycadopsida. So here this is the reason, because the plant have the thick, massive and short stem as like cycas. Leaves are large, frond-like and pinnately compound is also like cycas, and the seeds are radially symmetrical. So this all the character are related to the gymnosperm, particularly the cycas, and therefore the class of this plant is the Cycadopsida. Then the order is a Cycadophilical, or the another name is a Pteridospermale. So the Cycadophilical or the Pteridospermale, because this plant shows the both character, some character of the Cycadopsida and some character of the fern, and therefore the scientists give the name as a Cycadophilical. So Cycado is a part of the gymnosperm and filical is a part of the pteridophyte. So it is also called as a Pteridospermale, because the seeds bearing fern and it is placed in between pteridophyte and gymnosperm. So the Lyginopteris is one transition group between the pteridophyte and gymnosperm. So it have the megaspore, which have the small megaspore, and they have the curve shaped bracteate structure, reproductive structure, and which is the cupule is also found in this plant. And next one rank, that is the family Lyginopteridaceae, because the plants are climber and it is pinnately compound leaves. The genus of this plant is Lyginopteris and species is Oldamia, and the geological age of this plant is Upper Carboniferous to Permian rock, and the locality, that is England and the America. So here the discovery of the plant or history of the pteridosperm plant. So this Lyginopteris group is the first studied by Italian scientist Potoni in 1899. He discovered the medulla stem, leaves and petiole of this pteridosperm group. He named this group as a Cycadophilical. So here the Cycadophilical is the name given by the Potoni. So his observation, that means the Potoni observation is only on the observation of the vegetative character. Later on in 1903, the two scientists Oliver and Scott announced the discovery of the seed of the Lyginopteris, and he gave the name of this seed as Lyginostoma lomaxi. The scientists named this group as a pteridosperm. So here the Potoni give the name as a Cycadophilical, and the Oliver and Scott give the name as a pteridosperm. So here therefore this group is the transition group between the gymnosperm and pteridophyte. So this plant, that is a Lyginopteris plant, they have the organogenera, and therefore each and every part of the plant have the separate generic name. Here this is the generic name. The seed stem of the Lyginopteris is called as a Lyginodendron Oldamia or Lyginopteris oldamia. rachis or the petiole of this Lyginopteris plant is called a Lyginopteris aspera. The root is called a Callus hylar hookeri. The leaf is called a Spinopteris. The stamen or microsporophyll stamen-like structure, it is called as Crosophica, or it is called as a Telangium species. The megasporophyll, that is a megaspore bearing organ, is called as a Lyginostoma sinclairi. The seed of the Lyginopteris is called as a Lyginostoma, and the seed cupule is called as a Calamathistica hoengausi. So this is the generic name of different parts of the plant. So this is the Lyginopteris plant. This is the entire plant. So this plant is a climber, and it is a climb on the big woody tree of the forest. That means the stem is big, and therefore this plant is climb on the other support, that is the big tree in the forest. The stem is long, it is slender, weak and frequently branched, and the stem, the diameter is ranges from 2 to 4 centimeter. The stem is after maturation, it is aerial. Adventitious roots are also found here. So here this is the stem of the Lyginopteris. Then the plant are characterized by the frond-like leaf. So here this is the leaf, which is a frond-like, and it is looks like a pteridophytic leaf. The leaf genus is described as a Spinopteris. So this leaf is called as a Spinopteris. The leaves or the frond arranged on the stem, such a leaf was bipinnate or tripinnate. That means in some specimen there is a bipinnate leaves, and some specimen there is a tripinnate compound leaves, and at the base it is once forked, as well as at the apex here there is also forked structure we can observe here. The pinnae are born at right angle. So here this is the pinnae, this is born at right angle to the rachis and are oppositely arranged. The pinnules are or leaflets here, very small leaflets, very small and lobed. The leaves and the rachis are covered with the capitate gland, and this capitate gland is the diagnostic character of the Lyginopteris. When this capitate gland is a secretory in function. The plant produces the reproductive structure very similar to cycad. That means the external structure, that is a pinnately compound leaves, and the nature of the stem, the nature of the leaves, it is similar to the fern, but the reproductive structure, that is the megasporophyll, which is produced on the separate plant. That means microsporophyll, this is the microsporophyll, and megasporophyll, they are produced on separate branches, and this macrosporophyll and megasporophyll, they are similar to the cycas or to the gymnosperm. So here this is the fossil plant, and therefore there is a limited information is available, and here we have to discuss the transverse section of the stem. Already we have know the stem is not very much hard, and this stem shows the secondary growth. So here if you discuss the primary structure of the stem, then it have the epidermis, which is a single layered. Next to epidermis there is a broad cortex region. The outer cortex is broader in size and consists of the parenchymatous cells, in which radial patches of the sclerenchymatous cells are present, forming a vertical network of the sclerotic nest. The inner cortex is parenchymatous, which is soft, and they have the leaf traces are there. The vascular bundle is conjoint collateral open type. That means here this is a common vascular bundle, and due to this open type, this plant or this stem is open for the secondary growth, and the xylem is always a mesarch xylem. Due to secondary growth, the primary xylem here the pushes toward the pith region, and here in the pith region there is a primary xylem, as well as there is a sclerotic nest are also present. Sclerotic nest means it is a sclerenchymatous patches which are found in the pith region. So this part it shows the secondary growth, and this secondary growth because here this is the xylem cylinder, At the center here there is pith, which is small and which becomes small due to the growth of the secondary xylem. Here there is a secondary phloem, and this is the cortex region, which is crushed due to the secondary growth. That means here the secondary xylem consists of tracheids with multi-serried barbed pith. So here this is the characteristic of the stem of Lyginopteris or Lyginodendron, and this Lyginopteris stem, which is shows the advanced character. That means multi-serried barbed pith is the advanced character of this Lyginopteris. So here this is the caloxylon, that is the root of the Lyginopteris is described under caloxylon huckeri. When we take the transverse section of the root, then it is this root is adventitious, aerial, long, and it shows the typical vascular bundle, which is a radial type of vascular bundle, and they have the endarched xylem, and the large root also shows the secondary growth. So here this is the secondary growth of the root. The reproductive structure of the Lyginopteris. So Lyginopteris oldamia is heterosporous and heterothalic. Heterosporous means there is a micro and megasporous spores are present. Heterothalic means both, that is microsporangium bearing organ and megasporangium bearing organ, both are present on separate branch, therefore it is called as a heterothalic. The micro and megasporangium are present on separate branches and are always associated with the Spinopteris leaf. The reproductive structure also shows the capitate gland, which confirm their connection with the Lyginopteris. The microsporangia or the pollen producing organ has been described under the generic name called as Chrosotheca, and the megaspore bearing organ is described under generic name Lagistoma lomaxi. So first we go with the Chrosotheca, that is the microsporophyll of the Lyginopteris. The Chrosotheca was discovered by the Kidson in 1905 in the form of impression type of fossil. So here this is the branch, we call it as a Chrosotheca. It is an impression type of the fossil. It is a type of microsporophyll where the microsporangia are born on ultimate branches. So here this is the ultimate branches, and on this ultimate branches there is a, at the end of this ultimate branches there is a spoon shaped or spatulate shaped microsporangia there. So at the lower side of this spatula or this spoon shaped structure, 6 to 10 peripheral, so here peripheral hanging elongated microsporangia are present. Each microsporangia are very small, it is about 3 mm in long and 2 mm in breadth, and it is a bilocular. Inside these microspores are present, and this microspore shows the multicellular gametophyte. So here this is the called as a Chrosotheca, and it is ultimate branches. This is the pinna, and on this pinna at the end there is a hanging microsporangium are present. So here this is called as a Chrosotheca. The second specimen is the seed genus, or it is called as ovule. Actually it is in the form of the seed. In 1904 Oliver and Scott first time described this seed, and he gave the name as a Lagistoma lomaxi. That means this is the seed of the Lyginopteris oldamia, and it is described under generic name Lagistoma lomaxi. Each seed is a small barrel shaped and radial symmetrical structure. It is measure about 5 mm in length. That means it is small. These seeds are produced lateral or ultimate branches of the frond. The seeds are covered with curve-shaped structure. So here the seed is covered with the curve-shaped structure and which are covered with the cupule. That means the structure is covered with the capitate gland, and this cupule is, this capitate gland is again confirmed that this part or this seed is the seed of the Lyginopteris oldamia. Each seed in this contain orthotropous ovule with a single integument. So here this is the single integument and this is the orthotropous type of ovule, because the micropylon and this chalaza and this funicle, they are present on separate, they are present on same line or single line. Therefore it is called as a orthotropous ovule. So the integument, that is the envelope of the seed or covering of the seed, they have the two distinct layers called as a sclerotesta and endotesta. The external part is called as a sclerotesta, which is hard and stony, and inner fleshy endotesta, and in between the free part of the integument here there is a micropyle is there, and there is a one flask shaped structure. This is the flask shaped structure of the nucellus, is comes out, it is a prolonged structure, or this prolonged flask like structure is called as a Lyginostoma. So here this is the Lyginostoma of the seed, and therefore the scientists give the name as a Lyginostoma. And in 1944, the another scientist lab reported that the female gametophyte, so here this female gametophyte contain several archegonia in the ovule or inside this female gametophyte. So here this is the called as a Lyginostoma lomaxi. Second plant, which is not a plant but only organ, that is the fruit, dicot fruit or dicotyledonous fruit, called as Enigmocarpon. So the name indicates that enigmo is nothing but the problematic. So this fruit, their classification or their nature is a problematic. Therefore the scientists give name as a Enigmocarpon. So the classification of the Enigmocarpon is like this. So the first kingdom is the plantae, division is the Spermatophyta, subdivision Angiosperm, class Dicotyledons, subclass Polypetalae, family Lythraceae or Sonaceae. Two families are there. There is a controversy regarding the character. Therefore, some scientists have kept this fruit in Lythraceae and some other are kept in Sonaceae. Genus Enigmocarpon, and the name of the species is Parizai. So this is the recent fruit, that is geological age is the tertiary period, and the locality, that means this Enigmocarpon was found in intertrapine beds of Mohoga-Gallon in MP, India. That means this is the Indian fossil specimen, that is the Enigmocarpon. Then about this fruit. So this fruit, Professor Birbal Sahni in 1944 described first time, that is a dicot fruit under the generic name Enigmocarpon parizai. Due to its problematic nature, it was called as a Enigmocarpon. Enigmo is nothing but the problematic, carpon means fruit. The fruit is not problematic, but their nature is a controversial nature, and therefore it is a Enigmocarpon. According to Sahni, Birbal Sahni, it is a many-seeded, many-chambered, loculicidal capsule type of the fruit. So here this is the many seeds are there. This is capsule loculicidal fruit, and this is a type of the fruit is a capsule. He observed that the scientist Birbal Sahni observed that seed shows distinct character. So this seed shows a distinct character called as epistage. So the epistage is the small outgrowth which is coming from the nucellus, and this is the scientists call it as a epistage. Due to this character, the fruit was included under family Lythraceae. So the epistage like structure or epistage is present in the seed, and therefore the Birbal Sahni kept this fruit under family Lythraceae. Later on, the two scientists Mahabale and Deshpande in 1957 studied more specimens of the fruit and carried out comparative study of Enigmocarpon fruit and living fruit, as well as some other fossils fruit and living fruit. They state that epistage like structure is not a real epistage, not it is embryonic structure. They explained the structure found due to nuclear tissue which preserve against internal part of the epistage. And he kept this fruit under family Sonaceae. That means here in Sonaceae such structure is found, and therefore the scientist kept this structure or this fruit under family we call it as a Sonaceae. That means here some scientists it shows or it include under literacy and some include under Sonaceae. So here this is the Enigmocarpon dicot fruit.